This lecture is on contraction and excitation of muscle. We're going to look at a few different muscle types today. We're going to look at skeletal, cardiac, and visceral, sometimes also called smooth muscle. Ironically, the picture of skeletal here says cannot be displayed, but trust me, we're going to look at a lot of skeletal muscle pictures. Skeletal muscle, which is the focus of this lecture, is attached to the skeleton. And we know that muscles that are attached to our skeleton or to our bones are under voluntary control. Think about your biceps, your triceps, your muscles of your calves, your quadriceps. You can move these muscles voluntarily. You choose when to move them voluntarily. There's parts of your brain that are responsible for this. They connect conscious thought to movement, and you pick when to move them. If you look at these muscles under a microscope, they're striated. They have multiple nuclei, and they have They have fibers that run parallel to each other, and we'll look at a few of those pictures today in a moment. Cardiac muscle, which we'll talk about later this semester, and smooth muscle, which we'll talk about at the end of the semester, are different. They're under involuntary control. That means you and I don't control them voluntarily. We can't think and have it stop or start our heartbeat, or have it stop or start our digestion, although, of course, our thoughts are sort of tangentially related to and do have power over, through nerves and hormones, our cardiac cycle and our smooth muscles. We don't voluntarily control our heartbeat. So cardiac muscle is going to be a little bit different. It's going to be striated, so structurally it's going to look very similar and act very similar to skeletal muscle, whereas smooth muscle is going to be smooth. It's smooth microscopically. It doesn't have striations because it structurally is different. And each smooth muscle cell has one nucleus. Okay, it's just like a cardiac muscle cell does, whereas skeletal muscle have multiple nuclei. Biggest difference is, again, skeletal muscle attached to the skeleton under voluntary control. This is sort of what a skeletal muscle looks like. Like every cell, there's sort of some connective tissue and a membrane around the skeletal muscle cell, but each individual skeletal muscle cell is made up of a lot of different skeletal muscle fibers, these red things. Most of the weight of your skeletal muscle is actually skeletal muscle fibers. These fibers are the part of the skeletal muscle that actually do the mechanical work of contraction. These fibers have in and around them a cytoplasm, just like every other cell. We call the cytoplasm of a muscle cell actually the sarcoplasm. They have an endoplasmic reticulum, just like every other cell. We call that endoplasmic reticulum the sarcoplasmic reticulum. In this case, the endoplasmic or the sarcoplasmic reticulum is going to play a very important role in skeletal muscle contraction. The role it's going to play is it's going to be housing and releasing calcium. And it's actually going to be, as we've talked about before, calcium whose presence determine whether a skeletal muscle contracts or not. In the presence of calcium, a skeletal muscle would contract. When a skeletal muscle contracts, that's mechanical movement and work that the muscle does. When that skeletal muscle contracts, it becomes shorter. It shortens. So if we zoom in on one individual skeletal muscle fiber, it is composed of these individual contracting units called sarcomeres. This also gives skeletal muscles their striations. It appears to have striations. It has lines running up and down the muscles if you look at it under a microscope. And the predominant muscle fibers that actually do this contraction work are two types of muscle fibers, two proteins. They're called thin and thick filaments. And actually, to be a little bit more precise, they're called actin and myosin. You may have heard of these proteins before in other classes or in other contexts. Actin is actually a very predominant protein in a lot of cells of the body, if not almost all your cells. So in this picture, myosin is this red protein. Again, we're zooming in on the actual part of the muscle fiber that does mechanical work that contracts. These are the two proteins that interact for that contracting to happen. This red protein right here in the middle is called myosin. It's got these golf club-like heads on it that you see are trying to touch and interact with these yellow beads on a string that are called actin. Myosin is sometimes called the thick filament or the heavy filament. Actin is called the thin filament or the light filament. If we zoom in on them a little bit more, you'll see here that the thin filaments or actin are like strings on a pearl that are wound, and the thick filaments or myosin are like golf clubs that are sort of wound together, and then they have these heads. So these myosin heads want to interact with actin, and when they interact with actin in the presence of calcium, they're going to grab onto actin, they're going to pull this actin toward the center on both sides. Pull the actin toward the center until the actin runs into this midline. So that whole muscle fiber is going to move toward the center and the muscle is going to shorten. If you think about taking your bicep and flexing it like you're going to impress one of your friends, your bicep becomes shorter, which is what also makes it sort of bulge. Of course, it doesn't change in actual mass. It just reconfigures the shape of that bicep as you do mechanical work. Take your hands and open up your hands and put them right in front of you. Look at the palms of your hands, open up your fingers, and just move your fingers in and out so they interlock sort of with each other, like you're sort of grabbing your own hand and interlocking your fingers. That's sort of what happens. The myosin protein sort of grabs the actin and moves it towards the center and shortens the muscle filament so that the muscle does mechanical work. So again, here's that myosin wanting to grab onto actin. Now, it doesn't always grab onto actin because we know our muscle fibers aren't always contracting. So if we zoom in a little bit on actin, we'll see that actin has these little circles on it called myosin binding sites. So actin has a place on it to bind to myosin, but actin and myosin are not always bound. There are these proteins that have a regulatory role. So in addition to actin and myosin, there are regulatory proteins that control when actin and myosin bind to each other. These regulatory proteins are called troponin and tropomyosin. In this bottom graphic, they've been added and they've been superimposed in a realistic way on top of actin. And what they do is they block the myosin binding sites. They cover them so that at rest, a muscle has its myosin binding sites blocked, so myosin and actin are not interacting. There's two proteins that do this, troponin and tropomyosin. Tropomyosin is this long protein that is actually sitting and interacting with actin and blocking those myosin binding sites, whereas troponin is a calcium-specific or calcium-sensitive protein. So that when troponin is not bound to calcium, tropomyosin is blocking these actin binding sites. But when calcium comes around and is present in the cell, namely when you have an action potential, a cell flips its charge, troponin binds to the calcium that's now present in the cell. It moves tropomyosin off of those myosin binding sites that are covered on actin, and the myosin binding sites are exposed. Once those myosin binding sites are exposed, like is shown in this cartoon, then myosin is like, hey, it's on. I'm going to grab onto those myosin binding sites on actin. They're for me, myosin. And I'm going to, in the presence of calcium, pull that actin. In this picture, you'd be pulling it from left to right, towards the center, toward the midline. So these myosin molecules, when calcium's around, would grab actin and pull it toward the midline from left to right over here, and over here from right to left until that actin moved toward the center, toward the midline, and the muscle contracted. If you look at this picture, sort of with some electron microscopy, you'll see sort of a relaxed muscle, a partially contracted muscle, and then fully contracted muscle where these actin filaments have moved toward the midline. They're now actually overlapping each other. The muscle has gone from becoming longer to shorter. And so the muscle slides back and forth. This is how the muscle does mechanical work. We're going to add one more layer of detail on top of this, and that is the molecular details of how actin and myosin actually interact. You know this is mechanical work, so this is going to take some ATP. So we'll start with step one, which is ATP hydrolysis. For the myosin heads to actually become energized and to spring into a conformation where they can grab onto actin, they have to hydrolyze ATP. Again, ATP is adenosine triphosphate. It's an adenosine molecule with 3 phosphates. When it becomes hydrolyzed, it loses a phosphate and it becomes ADP. Usually that phosphate ends up kind of floating away in the cell, but what that allows myosin to do is then become charged, quote unquote, and grab onto actin. So step number 2 is attachment. When that ATP is hydrolyzed, myosin is charged. It's in the right configuration to grab onto actin. Once it grabs onto actin, it's game on. It then does what's called the power stroke. It has bound itself to actin and it pulls actin toward itself, toward the center. It yanks actin toward the middle. Then it detaches. After it detaches in step 4, it grabs onto another ATP, and it does step number 1 again. Hydrolyze that ATP to charge, to grab, to pull it toward the center, to power stroke it, and then to let go. Essentially, the myosin heads are just constantly in the presence of calcium, grabbing actin, pulling it toward the center, grabbing it, pulling it toward the center, grabbing it, pulling it toward the center until the muscle is maximally contracted, as is sort of shown here in this cartoon. This is an overview again of that sliding filament mechanism.